Hello everyone. In this laboratory, we'll observe the histology slides of circulatory system. Objectives: understand and identify the light microscopic structural characteristics of the main organs of heart, large artery, medium-sized artery, and medium-sized vein, small artery, and small vein. The first slide is heart wall, obtained from the section ventricle of heart. The light micrograph on the left, the uneven surface of the specimen is endocardium. The thick central part deeply stained in myocardium. The even outside surface stained weakly is epicardium. Epicardium is slightly thicker than the endocardium, consists of connective tissue, which contains blood vessel, nerves, lymphatic vessel of the pericardium, but sometimes has dropped. Endocardium is a thin layer lined by a layer of light endothelium cells. Outside the endothelium is subendothelium made of a thin layer of connective tissue, which contains fine collagen fibers, elastic fibers, fibroblasts, and some smooth muscle. Green arrow indicates between the subendothelium and the myocardium is the subendocardial layer of connective tissue, which contains Purkinje fibers. Yellow star indicates Purkinje fiber is thicker and stained light than cardiac muscle. The cytoplasm is abundant and around the nucleus. Myofibrils are near the cyclolemma and less than those in the cardiac muscle. 1 to 2 nuclei, stained slightly violet blue, are seen in the center of the Purkinje fiber. Myocardium occupies the most of thickness of the heart wall and consists of cardiac muscle fibers arranged in the spiral orientation, so longitudinal, transverse, and oblique section of cardiac muscle can be seen in the specimen. Red arrow indicate a lot of blood vessels are among the cardiac muscle fibers, such as capillaries. They have a small lumen enclosed by 2 to 3 endothelium cells, sometimes even only one cell, forming a ring-shaped lumen. In the light micrograph on the right, heart valves consist of dense connective tissue, leafless, covered by endothelium, that allow passage of blood only in one direction. Blue arrow indicates the left atrioventricular valve has 2 such leaflets, whereas the right AV valve and both valves from the ventricle has 3. The valve edges normally fit together closely when closed, preventing black flow of blood. The dense connective tissue of each valve leaflet is continuous with a large mass of dense connective tissue at the base of valve. This fibril is skeleton. The structure of heart valve may be slightly abnormal because of development defect. Scarring after infarction or heart problem. The second slide is large artery. In the section, the largest vessel, appearing as a half circle, is large artery. It's divided into 3 tunicles, from inside to outside. A very thin layer next to the lumen is tunicula interno. The tunicula media of large artery is thickest part of the wall. About two-thirds of the entire thickness is formed by several dozens of layers of strong, refracted, purple-stained elastic membranes such as the micrograph on the right with special staining. And between them, there are some smooth muscle, elastic, and collagen fibers. The tunica adventitia is relatively thin, but thicker than the intima. There are some fat cells, vasa, and bundles of nerve fibers in the connective tissue of the tunica adventitia. As both internal and external elastic membranes are in contact with the elastic membranes of the media, not as clear as those in the medial-sized artery, there are no distinctive dividing line between three tunica. The third slide is medial-sized artery and medial-sized vein, obtained from the section of medial-sized artery and medial-sized vein with HE staining. There are two medial-sized vessels paralleling each other, one which is deeply stained red and has thick wall and regular round lumen, is medial-sized artery. Black star indicates another, which is slightly stained and has thinner wall and relatively large, irregular, slightly collapsed lumen, is medial-sized vein. Blue star indicates. Around the medium-sized vein are connective tissue in which there are small arteries and veins. The wall of the medial-sized vein is thinner than that of the medial-sized artery. It is also divided into three tunica, but dividing lines are less obvious than those of the medial-sized artery. Both internal and external elastic lamellae are not obvious. The tunica media is quite thin and consists only of 3 to 5 layers of concentrically arranged smooth muscle. The tunica adventitia is quite thick and consists of abundant loose connective tissue with a few elastic fibers, longitudinal smooth muscle, vessel viral, and nerve fibers. We can see the structural characteristics of medial-sized artery in the next slide. The fourth slide is medial-sized artery, obtained from the section of medial-sized artery with HE staining. Differentiate the three tunica of the medial-sized artery from inside to outside. A very thin layer next to the lumen is tunica interna. Innermost layer of tunica interna is composed of squamous endothelium cells, the contour of which is not clearly visible. The nuclei of endothelium are fusiform or ovoid, slightly protruding into the lumen. Next to the endothelium is subendothelium layer, that's very thin layer of slightly stained connective tissue. Between the tunica intima and the tunica media, a layer of veil membrane, red in stain with strong refraction, is called internal elastic membrane, which is composed of elastic fibers. Black arrow indicates. The center of artery wall is tunica media. That's the thickest one of the three tunica, and consists of many layers of smooth muscle in circular arrangement. Some nuclei twist like a spiral because of the contraction of smooth muscle fibers. Some pink, wave elastic fibers with high refraction can be seen between muscle fibers. Collagen fibers are lightly stained and not easy to identify. The outside layer is tunica adventitia, which is made up of connective tissue. Near the tunica media is the external elastic membrane composed of many layers of elastic fibers, which forms the boundary line separating the tunica media from the tunica adventitia. Blue arrow indicates. The elastic fibers are cut into irregular fragments, which become sparser outward and finally can't be identified from the outside loose connective tissue. There are small vessels and nerves in the adventitia. The last slide is small artery and small vein, obtained from the section of small artery and small vein with HE staining. Small arteries are small and round with thick walls. Number 1 indicates the inner elastic membrane is thin, and the tunica media has several layers of circular smooth muscle. The small vein has a slightly large lumen and thinner wall than the corresponding small artery. Number 2 indicates its lumen is often collapsed and irregular for less of elastic component. Outside the tunica intima, there are some loose, arranged smooth muscle in the connective tissue. The tunica adventitia is relatively thick. Both internal and external elastic lamella are absent. Okay, that's all for today. Thank you.